for today because of her personal problems. Somebody is critically ill close to her family. And in place of Anupama ma'am, today from our institution, Dr. K. Naveen Babu sir is going to present a topic on biomolecules from actinomycetes. I hope you can understand the situation. And we are going to proceed with Dr. K. Naveen Babu sir as a resource person for today's session. I heartily welcome the esteemed speaker of today's session from our own institution, from the host institution, Dr. K. Naveen Babu sir, Professor. Sir is going to deliver a topic on biomolecules from actinomycetes. So let us listen to the brief profile of Dr. K. Naveen Babu sir. Now I request Dr. K. Ravishankar sir read out the profile of
Thank you,
Naveen Babu sir. K. Naveen Babu sir.
So myself, Dr. K. Ravishankar, Assistant Professor of Department of Pharmaceutics and Biotechnology. I take this privilege to introduce our beloved Professor, Dr. K. Naveen Babu sir, Professor of Department of Pharmaceutics and Biotechnology at KVSS Siddhartha College of Pharmaceutical Sciences. Dr. Naveen Babu sir completed B.Pharm from TS College of Pharmacy, Bengaluru, M.Pharm from AE College of Pharmaceutical Sciences, Andhra University, Visakhapatnam, and PhD from Central Food Technological Research Institute, Mysore. Sir is having a total of 19 years of experience in teaching and research. He is having a vast experience in research and also in the teaching. Sir has published more than 71 publications in peer-reviewed journals, most of which are indexed in Scopus and Science papers. Sir has got 2 patents to his credit, and Sir has guided more than 6 PhD students. Sir is expert reviewer of more than 15 scientific journals.
Hello.
Sir is expert reviewer of more than 15 scientific journals. Sir has authored 2 book chapters, and Sir organized more than 12 seminars and gave more than 9 guest lectures at several institutes. Sir has got membership at several professional bodies, and Sir is a member of Board of Studies in Pharmacy, Krishna University. With this brief introduction, I request our beloved Professor K. Naveen Babu sir to address this session. Sir, Naveen Babu sir.
Thank you very much, Dr. Ravishankar. Good afternoon to all. Today I am going to discuss about the bioactive molecules from actinomycetes. Microorganisms are among man's best friends and worst enemies. Exploitation of microorganisms for useful purpose is known since 6000 BC. History reveals many applications of microbial process for the production of desirable materials. The hastening of activities of microorganisms represents one of the most fascinating aspects of man's scientific and technological development. From the standpoint of industrial microbiology, microorganisms can be considered as chemical factories in miniature. Okay. They have the potential to produce novel and new therapeutic agents, to convert relatively inexpensive raw materials into end products of value for human use, thus becoming attractive for commercial exploitation. Actinomycetes are that group of intracellular branching organisms which reproduce either by fission or by means of spores or conidia. From an ecological point of view, Actinomyces stand an intermediate position between the fungi and the bacteria in terms of numerical frequency of occurrence in various biotypes. They are closely related to the true bacteria and frequently they are considered as high filamentous bacteria. The outstanding fungal characteristics of okay. The outstanding fungal characteristics of actinomyces is morphological position of true branching mycelium. In addition, actinomyces may also show strong parallels with the true fungi in their production of sporangia and motile spores. However, mycelium diameter and spore size is of lower order of magnitude in actinomyces, averaging from 1 millimeter only as compared with fungi. They are usually form a mycelium which may be of a single kind designated as substrate or two kinds of substrate and aerial mycelium. They produce wide variety of spore types which include endospores, long regarded as the typical spore structure of eubacterials. Some genera such as Streptomyces and Micromonospora form an extensive branched mycelium composed of individual hyphae subdivided by frequent cross walls. The actinomyces are predominantly aerobic, heterotrophic, and saprophytic. They have a cell wall structure characteristic of bacteria and frequently show the presence of lytic viruses. It is estimated that 85% of the antibiotics from microbial sources are being produced by actinomyces. This is the main reason to take the topic of today's. Since the discovery of streptomycin by Waxman in 1943, interest of many microbiologists was switched over to explore naturally occurring substrates for isolation of novel actinomycin strains and novel metabolites with resounding commercial success. This is the main reason for prominence of conventional screening programs even at this stage. Many biological molecules are known to inhibit specific enzymes also. As several diseases are associated with abnormal enzyme activities, this concept has yielded valuable to pharmaceutical compounds. They can be also known as target enzyme inhibitors and display distinct pharmaceutical action depending on the target enzymes. The screening of microbial culture filtrates for low molecular mass enzyme inhibitors initiated by Hamo Umezawa in 1966. The first inhibitor, lupeptin, was discovered in 1969 as a metabolite of Streptomyces strain. Since then, many inhibitors have been discovered in culture filtrates of actinomyces, bacteria, fungi, and some have been introduced commercially. Coming to the how to how to define the targets. Here in in this condition, here the target is unknown, the microorganism is known, and the molecule produced from that microorganism is also known. Means when we do regularly the isolation of microorganisms from the soil sample or water soils, it is a continuous process. And after the isolation, the production of the compounds will be done. If the compound is having activity, high activity for anyone, we are going to do the structural analysis, and by that time we'll be knowing the name of the molecule. If it is a new one, we are going to name it, or if it is the old one, we will know the molecule. But these cultures and the compound extracts will be stored, and they will be checked by from time to time. For example, all the for the coronavirus, almost almost all the compounds antibiotics which are present were screened for the coronavirus. In the same manner, we are going to do the screening process for every new disease or for new organism, for every new pathogenic microorganism. And the next one is here the target is known, the microorganisms are known, but the molecules are unknown. In the third one, it will be the target is known, but the microorganisms are unknown and molecules. Means here, for example, here we need an antiviral drug, that is our target, but right now we don't know from which organism we are going to get the molecule, and even the molecule is unknown. Initially, the screening will be done without knowing the microorganism and even the molecules. Generally, the source of the microorganisms will be terrestrial or aquatic environment or marine. Generally, collection of the soil samples will be dependent on the organic matter, moist content, particle size, color of the soil, geographical distribution. The most important is here the geographical distribution, because here if we are going to collect the samples very nearby, the organisms that may come into our petty plates might be the same. That's why we have to take care for all these, that is organic matter content, moisture content, particle size, color of the soil and geographical distribution. How we are going to isolate these organisms from the soil sample? Initially, we are going to collect the soil sample in a sterile container by removing the upper layer of the soil about 1 inch, 1 to 3 inches, and from there we are going to collect the soil sample into the sterile container. The sterile container is brought to the lab, and here we may do the pre-treatment for soil samples for the desired microorganisms. The pre-treatment might be heat treatment, centrifugation, calcium carbonate, or phenol. Superficially, for if it is for the streptomyces, we are going to use the calcium carbonate treatment. Here we are going to take 10 grams of the soil sample was ground in a mortar with calcium carbonate in the ratio of 1:1 and incubated for 10 days at 28 degrees centigrade in a closed inverted sterile petri dish with water saturated filter paper dish. From this, 2 grams of the sample was taken into a 100 ml of sterile distilled water and agitated by shaking on a rotary shaker at 220 rpm for 30 minutes at 28 degrees centigrade. After the shaking, we are going to keep the flask aside for another 30 minutes such that the soil matter will be settled down and the supernatant was then taken and serial dilution was done. In the same manner, if it is for the phenol treatment, 10 grams of soil was mixed with 100 ml of sterile distilled water within a 500 ml Erlenmeyer flask and agitated on a rotary shaker at 220 rpm for 5 minutes at 28 degrees centigrade. This suspension was then mixed with 100 ml of 1.4% weight by volume of phenol solution and incubated for 10 minutes. The supernatant was serially diluted with sterile distilled water. In case of dry heat treatment, if we are going to isolate the microorganism which are in the form of spores, we are going to treat in this method. 10 grams of soil sample was treated at 100 degrees centigrade for 1 hour. Of these, 1 gram of sample was then mixed with 100 ml of sterile distilled water, and it's the same procedure to be repeated. Means here, if we are if our desired organism is an spore forming, we can heat it at 100 degrees centigrade for 1 hour. Means all the microorganisms present in the vegetative form will be killed by this treatment. The other method is by centrifugation. 10 grams of the sample was mixed with 100 ml of sterile distilled water in a 500 ml Erlenmeyer flask and agitated on a rotary shaker for 5 minutes at 28 degrees centigrade. The soil suspension was centrifuged for 20 minutes at 6000 rpm. The supernatant was then serial diluted with sterile distilled water. Once the treatment of the soil sample is over, the first step will be the serial dilution. Why we are going to do the serial dilution here is to grow the to get the discrete colonies in the petit plate. Means if the number of colonies will be more, the colonies will be side by side, and we cannot pick uh, these colonies because they will be messed up with each other. Later, we have to do the isolation, and here we are going to take the media. The media will be sterilized at 121 degrees centigrade for 15 to 20 minutes. After sterilization, the media is cooled down to 40 to 45 degrees centigrade. To this media, we are going to add 1 ml of the serial diluted suspension and mix thoroughly and poured into a sterile petit plate. And these plates are incubated at 28 degrees centigrade for more than 21 days. In the pour plate method, the mixed culture is diluted directly in cubes of liquid agar medium. A series of agar plates showing decreasing number of colonies resulting from the dilution. The pour plate technique has certain disadvantages. For instance, some of the organisms are trapped beneath the surface of the medium in the gel form. Therefore, both surface and subsurface colonies develop. The subsurface colonies can be transferred to a fresh media only by first digging them out of the agar with a sterile instrument. Another disadvantage is that the organism being isolated must be able to withstand temporary exposure to 45 degrees centigrade. That is, the psychrophilic bacteria cannot be isolated by this method. But whereas in the case of spread plate method, this can be done because in the spread plate method, the mixed culture is not diluted in the culture medium. Instead, it is diluted in a series of tubes containing a sterile liquid, usually water or physiological saline. A sample is removed from each tube, placed onto the surface of an agar plate, and spread evenly over the surface by means of a sterile bent glass rod. Or, in a simple way, from the serial dilution, we are going to add a drop or two drops on the agar surface, and this is spread with an sterile bent glass rod. On at least one plate of the series, the bacteria will be in numbers sufficiently low as the development of well separated colonies. In contrast to the pour plate technique, only surface colonies will develop. Moreover, the organisms are not required to withstand the temperature of liquid agar. Unlike the streak plate technique, the pour and the spread plate techniques may be performed for isolation of microorganisms. This is the figure for how we are going to do the serial dilution. Initially, we are going to sterilize the media at 121 degrees centigrade for 15 to 20 minutes. The medium will be cooled to 40 to 45 degrees centigrade, and from each dilution, we are going to add 1 ml to the medium, rolled between the palms for the uniform mixing of the culture suspension, and poured into a sterile petty plate and incubated at 28 degrees centigrade for 21 days or 3 weeks. This is the way we are going to do the spread plate method. And here, what are the different media that are used for screening of actinomycetes? For screening of actinomycetes, we are going to use chapak drops, egg albumin agar, glucose asparagine agar, half strength nutrient agar, starch casein agar, nutrient agar, raffinose histidine agar, CYC agar, gauze agarized medium number 1, Benedict's medium, Liden beans medium, humic and vitamin agar, potassium tellurate, AV agar, or oatmeal agar. After 21 days of incubation, we are going to get the colonies in the petty plate in this manner. After the growth of the microorganisms, we are going to subculture these microorganisms into glycerol asparagine agar. The composition is L-asparagine 1 gram, glycerol 10 grams, dipotassium hydrogen phosphate 1 gram, trace salt solution 1 ml, back to agar 20 grams, distilled water quantity sufficient to 1000 ml, and pH of the medium A will be 7 to 7.4. While subculturing the isolates, we have to take care of by looking by the naked eye, that is, what is the color of the mycelium, what is the reverse color of the colony, what is the soluble pigment, what is the texture of the colony, and then we are going to subcultures from the petty plate to slants. Why we have to take this care is for unrepetition of the same microorganism for subculturing. Even we are going to perform the macro and micro morphological features of the colonies developed on various media, and the color determination of the aerial mycelium, substrate mycelium, soluble pigment, these all were examined after subculturing to the fresh tubes after 14 days of incubation. The macromorphology was noted by the naked eye. The colors of the aerial mycelium and substrate mycelium soluble pigment when grown on different media were observed and recorded. Micromorphology was observed by placing the sterile cover slips at an angle of 45 degrees centigrade in solidified agar medium in a petri dish such that half of the cover slip was dipped in the medium. Inoculum was spread along the line where the upper surface of the cover slip meets the medium. After complete sporulation, the cover slips were removed and examined by phase contrast microscope. Colors were matched with one of the seven color wheels of Fresnel and Bacchus. Generally, we are going to check these macro and micro and macro morphological features on the medium like tryptophan, yeast extract broth, yeast extract, malt extract agar, oatmeal agar, inorganic salt starch agar, glycerol asparagine agar, peptone yeast extract agar, tyrosine agar, Bennett's, nutrient, Chapak dogs, and modified Bennett's agar. After subculturing of the microorganisms into the test tubes, they will be in this manner. Before subculturing, we have to take care of all the what all we have discussed till now. After the subculturing, we have to ferment these cultures. Again, the fermentation might be done on solid state fermentation or submerged fermentation. Means we are going to add the microorganism to the medium. For actinomycetes, solid state fermentation is the best. For fungi, the submerged fermentation is the best. Actually, if the microorganism goes on the submerged fermentation, the cost reduction will be very less in solid state fermentation, because in the solid state fermentation we are going to use almost the raw material cost is very low, like maize flour, starch flour, even we can use all these, and the cost will be more effective when compared to solid state fermentation. Generally, after the fermentation, we are going to extract it with solvents or with aqueous broth filters. In generally, we prefer for the solvents. Here, if the compound is known or if it is the desired molecule, we know to, with which with which solvent we have to extract, or we know the solubility of the molecule beforehand and we are going to use the same solvent. If it is in the case of unknown compound, we are going to use ethyl acetate, because in ethyl acetate both polar and non-polar compounds can be extracted. So for unknown compounds, we generally use the solvent extraction as ethyl acetate. Next, here the after extraction, we are going to check for both primary screening and secondary screening. Either whatever it is the activity may be checked through for the target. The target might be antiviral, antibacterial, antifungal, or any enzyme inhibitor. These enzyme inhibitors are generally can be used as lipase inhibitors. Lipase inhibitors are nothing but anti-obesity, glucose 6 phosphate. Okay. Anyhow, I'm going to discuss that at the end. Okay. The overall outflow of the chart till now, what we have discussed will be this. Finally, after the extraction of the solvent, we are going to distill it under reducer pressure. The crude extract, if it is for enzyme inhibition, the crude extract will be dissolved in DMSO because DMSO does not inhibit any enzyme. But the solvents like ethyl acetate, chloroform, ethanol, methanol, they'll be inhibiting the enzyme. So if it is for enzyme inhibitors, we are going to dissolve the crude extract in DMSO. And if it is for antibiotic assay, we can dissolve it in any solvent or even in aqueous solutions. If it is in aqueous solution, we are going to perform the assay by couplet method. And if the compound is soluble in any solvent like chloroform, it's ethyl acetate, we are going to perform it in disc plate methods. After the crude extraction, after the crude extraction, we are going to check it for antimicrobial activity and enzyme inhibitors. First, initially, we'll see how we are going to perform the assay of the antibiotic. It can be done in the same manner for any for virus, even bacteria or fungi also in the same manner. The main principle of the microbiological assay of antibiotic is comparison of the inhibition of growth of microorganism by measured concentration of the antibiotic under examination with that produced by known concentration of a standard preparation of the antibiotic having a known quantity. Right here, generally it will be of two methods. One is the diffusion method, the other is turbidometric method. Right? Initially, we have to check our crude extract with different microorganisms, the activity against different microorganisms. After means to which the antibiotic is highly sensitive. After, for example, here we have checked the crude extract with n number of microorganisms, and the the crude extract is highly sensitive towards Bacillus subtilis. Then we are going to take streptomycin as the standard, because in the principle itself, in a simple manner, we are going to we are we are going to compare the potency of our sample with that of standard. To select the standard antibiotic, we have to initially perform the assay with different microorganisms. After performing, we have to check to which organism the microorganism is highly sensitive. Depending upon the sensitivity of the microorganism, we are going to select the standard antibiotic. Here, if it is in this slide, we can see if it is for Bacillus subtilis, we can choose streptomycin as standard. After choosing streptomycin, this is all from IP only, right? After choosing streptomycin as standard, here it is given assay method as A and B. Assay method A is nothing but diffusion method. Method B is turbidometric method. In the second column, we can see whether the streptomycin has to be dried prior or not. Here it is given as S. In which solvent the streptomycin has to be dissolved, that is water, and what is the final stock concentration per ml is 1 mg per ml. After the preparation of this stock solution, we can use this for 30 days. From this stock solution, we are going to prepare different dilutions. And how we are going to prepare the different dilutions is, even the median concentration was given in the IP, that is the middle one. Here it is, by for method A it is 1 microgram per ml, and for method B it is 30 micrograms per ml. Generally, for couplet methods, we are going to use around 25 ml. In this case, 25 micrograms will be the median concentration. To draw any standard curve, we should have at least 5 points. Here, the middle concentration is 25 micrograms. Now we have to make two dilutions up and two dilutions below, that is S1, S2, S3, S4. S3 is already given. We have to prepare for S1, S2, S4 and S5. These dilutions must be prepared in the ratio of 1 is to 1.25, right? Here we have to separate the 25 ml of media into two parts. The first part will be 21 ml, and the second part will be 4 ml. In generally, the microorganism is added to the 4 ml medium. Before that, we have to standardize how much ml or how much inoculum should be added to the medium. Here initially we have to grow the microorganism at 28 degrees centigrade for if it is bacteria, we have to grow it for 5 consecutive days. Means the culture should be, if it is from the lab, we have to take the lab culture, we have to inoculate that into a fresh medium and incubate it for 16 to 24 hours. After the incubation of 16 to 24 hours, from there we have to transfer the inoculum into the fresh medium. In the same manner, we are going to subculture the bacteria, if it is bacteria, by 5 consecutive days, and while doing the assay, the culture should be less than 24 hours old. Means in generally we perform it around 16 to 24 hours period. Later on, after 5 consecutive days of culture, it should be less than 24 hours old culture. Now we are going to centrifuge it for 3 times and resuspend it for 3 times. If the microorganism is grown in the broth, when we centrifuge for the first time, there might be some macromolecules present in the medium which will be suspended along with the microorganism. To remove these macromolecules, we are going to resuspend for 3 times with sterile saline water or sterile distilled water. After the resuspension, after the third centrifugation, now we have to go or we have to measure at the spectrophotometer or calorimeter at 530 nanometer. We have to make whatever is the dilution, we have to measure the optical density. Even we can fix the optical density here initially. After fixing the optical density, this culture suspension was added to the medium. Means different aliquots of this culture suspension is added to 25 ml of media and incubated in the incubator and for 16 to 24 hours. After that, now we have to check in which petty plate the growth of the microorganism is uniform throughout the plate, or simply we can say in which plate we got the lawn culture. Now we have to fix this to the assay medium. That means by at 530 nanometer, if the culture suspension what we have taken is 0.5 OD, and from this 0.5 OD, if after adding 2 ml to the culture to the media, if the growth is uniform throughout the uniform on the agar surface, now we have to fix 530 nanometer, 0.5 OD and 2 ml as the inoculum to the assay medium, right? Next, the method is, in generally, we have to take around 15 petty plates, in which 12 petty plates will be for standard and 3 petty plates will be for our unknown compound. Here again I repeat, the principle is comparison of unknown with that of standard. Right now we are comparing our unknown compound with the standard. Here in each group there will be 3 petty plates. The groups are only 4, that is S1, S2, S4 and S5. There will be no group for S3 because here S3 is added in every alternative cup, and in each petty plate there will be 6 cups. These cups will be cut with an cork or borer with with an outer diameter of 8 mm or inner diameter of 6 mm of the cork, right? The next thing is we have to add the antibiotic solution to the cups. After adding the antibiotics to the cups, we are going to place these petty plates in refrigerator at 4 degrees centigrade for the diffusion of the antibiotic. The diffusion of the antibiotic can be carried out at the room temperature, but the problem here arises is the growth of the microorganism starts. To inhibit the growth of the microorganism, we are going to place the petty plates at 4 degrees centigrade. After the complete diffusion of the antibiotic solution into the agar, we are going to place these petty plates in incubator in an inverted position. That is, generally the bottom will be on the upper side and lid will be down in the inverted position. After incubation for 16 to 24 hours, there will be a zone of inhibition as shown in the slide. After, now we have to measure all the diameters of zone of inhibition of all in all the petty plates. After the measurement, we are going to get 36 values in the 36 values of median concentration, because we are taking 12 petty plates. In each petty plate, there will be 6 cups, and in each petty plate, we are going to add alternatively the median concentration. So, after taking the measurement of the median concentration of the 36 zone of inhibitions, we have to take the average or mean. Now, this average and mean will be the standard for us. Again, we have to come back to the S1 group. In the S1 group, there are 3 petty plates, and in each petty plate, there are 3 S1 values and 3 S3 zone of inhibition values. That means in the total, we are having 9 values of S1 and 9 values of S3. Now, with the 9 values of the S3, we are going to take the average and compare with these 36 value. If the value is the same, there will be no correction factor. If the value is different, we have to make the correction factor. For example, if the 36 values mean is 20 mm, and if it is in the case of in the S1 group, if it is S3 mean value is 19, then we have to add +1 to the S1 mean values. That is, we have to calculate the S1 9. We have to measure the S1 9 zone of inhibitions and take the mean or average value. If it is, for example, S1 mean value is 6, we have to add this correction factor +1 to that, and finally, after the correction factor, the value of S1 will be 7. In the same manner, vice versa, if it is more, we have to minus from the S1 mean or average value. That is, if, okay, we'll see if it is in the second case, if it is S2, in S2 case, the S3 median concentration average or mean is 21, then we have to minus 1 in the S2 9 average zone of inhibition values. In this manner, we have to do the correction factor for all. Even parallelly, we are going to check the zone of check for the unknown. For unknown, we have taken 3 petty plates. Here also, we are going to add, along with the unknown, we are going to add the median concentration alternatively, and we have to cross check the median for the median values with that of the standard 36 values. If any correction factor is there, we have to do. If there is no correction factor, we can take directly the value of 9 values of the unknown directly, the average and mean, and now we have to make the graphical representation. In the graphical representation, we are going to draw the graph for the 5 values. If that is like a straight line, at least more than at least 3 points have to correlate with this line. The other should be 1 point should be above on the other point should be below. Now now we are having the unknown zone of inhibition value. From the y-axis, we have to extrapolate to this line, and from there we have to interpolate to the x-axis. Now, how we are going to say the potency of the unknown or potency of our drug is with the comparison, the unknown concentration of dash, that might be micrograms or milligrams, is equivalent to this at this point where it has touched, it is the standard that will be, for example, if it is streptomycin, it will be in these many micrograms of streptomycin. In the same manner, we are going to perform the displate method also. The method will be the same. Here, the method in displate method, we are not going to cut the cups or cups in the media. Here, instead of cups, we are going to take Wattman filter paper number 1 and punch, and we are going to add the antibiotic solution to these discs. Adding means we are not going to add, we have to just moisten the disc with the antibiotic solution. The main difference between the cup and disc plate method is the extracts which are soluble in aqueous solution can be done by couplet method. The extracts which are soluble only in solvents must be done with disc plate method. After adding the antibiotic solution to the disc, we are going to impregnate this disc on the agar surface. Here also, for each petty plate, we are going to add 6 discs, and the alternate will be the median concentration. Same, it is same as that of couplet method. Even the estimation of the potency is also same as that of couplet method. Next, coming to the turbidometric method. Here, the ml of media will be 10 ml. So, as we have seen before, if it is streptomycin, the median concentration is 30 micrograms per ml. Here, the median concentration will be 300 micrograms, and we have to calculate S1, S2, S4, S5 in the ratio of 4:5. Right? Here, the potency, we are going to take the medium in a broth. Not, we cannot perform this assay in agar. In agar, here we are going to perform it in the liquid medium. We are going to take 9 ml of medium plus 1 ml of antibiotic solution. To the 9 ml of media, we are going to add 1 ml of antibiotic solution and microorganism to the medium, and we are going to check the potency with the spectrophotometer at 530 nanometer and take the optical density values. Here also we are going to perform in the graphical method, that is concentration of antibiotic versus optical density. In the before case, it is concentration of the antibiotic versus zone of inhibition in millimeters. Right? Here we have to take at least 3 controls. The first control will be after sterilization. We are not going to add anything. This is for sterility testing, but this tube is incubated along with the other test tubes. The second control will be media plus microorganism. This is for growth promotion test. We have to check whether the microorganism grows very well in the medium or not. The third control will be medium plus microorganism plus 0.5 ml of 0.5% formaldehyde. Here we are going to take 9 ml of media plus 1 ml of antibiotic solution, plus we are going to add the microorganism and incubate for 3 to 4 hours only. Whereas in the case of couplet method or displate method, we are going to incubate it for 16 to 24 hours. Here in turbidometric method, it is only 2 to 4 hours. After the incubation, we have to take the optical density at 530 nanometer. And the other way of estimation of the potency is L is equal to 3A plus 2B plus C minus E by 5. That is nothing but what here before I have told you in the form of S1, S2, S3, S4, S5. Here it is A, B, C, D, E, F. That is 3S1 plus 2S2 plus S3 minus S5 by 5, and H is 3S5 plus 2S4 plus S3 minus S1 by 5. This is the other way of estimation of the potency. L is the calculated zone diameter for the lowest concentration of the standard curve response line. H is the calculated zone diameter for the highest concentration of the standard curve response line. C is the average zone diameter of 36 readings of the reference point standard solution. ABD is corrected average values for the other standard solutions, lowest to highest concentration respectively. And we have to select the media for the particular microorganism from the IP only. Here, for example, if it is Bacillus subtilis, it is A, and here in this table we can see the composition of the medium for A. The same medium can be used for both method A or method B, that is couplet or discrete method or turbidometric method. In turbidometric method, we should avoid agar. The same crude extracts, whatever we have isolated, can be used for the enzyme targets. Enzymes are excellent targets for pharmaceutical intervention owing to their essential roles in life process and pathophysiology. The structure of enzyme active sites and other ligand pockets on enzymes are ideally suited for high affinity interactions with drug-like inhibitors. Not surprisingly, enzyme inhibitors represent almost half the drugs in clinical use today. Recent surveys of the human genome suggest that the proportion of the genome that encodes for disease associated druggable targets is dominated by enzymes. More than 40% of the drugs now are enzyme inhibitors. An enzyme inhibitor is a molecule that binds to an enzyme and decreases its activity. By binding to enzymes, active site inhibitors reduce the compatibility of substrate and enzyme, and this leads to the inhibition of enzyme substrate complex formation, preventing the catalysis of reaction and decreases the amount of product produced by a reaction. It can be said that as the concentration of enzyme inhibitor increases, the rate of enzyme activity decreases, and thus the amount of product produced is inversely proportional to the concentration of inhibitor molecules. Since blocking an enzyme activity can kill a pathogen or correct a metabolic imbalance, many drugs are enzyme inhibitors. They are also used in pesticides. Not all molecules that bind to enzyme inhibitors are enzymes are inhibitors. Enzyme activators bind to enzyme and increase their enzyme activity, while enzyme substrates bind and are converted to products in the normal catalytic cycle of the enzyme. Generally, enzyme inhibitors are classified into reversible and irreversible. Reversible inhibitors attached to the enzyme with non-covalent interactions such as hydrogen bonds, hydrophobic interactions, and ionic bonds. Multiple weak bonds between the inhibitor and the active site combine to produce strong and specific binding. In contrast to substrates and irreversible inhibitors, reversible inhibitors generally do not undergo chemical interactions when bound. When bound to the enzyme and can easily removed by dilution or dialysis. There are four kinds of reversible enzyme inhibitors. They are classified according to the effect of varying concentration of enzyme substrate on the inhibitor: competitive inhibition, uncompetitive inhibition, and non-competitive inhibition, and mixed inhibition. The first one is the competitive inhibition. The substrate and inhibitor cannot bind to the enzyme at the same time. This usually results from the inhibitor having an affinity for the active site of an enzyme where the substrate also binds. The substrate and inhibitor compete for the access of the enzyme active site. This type of inhibition from the inhibitor having affinity affinity for the active site of an enzyme where the substrate also binds. The substrate and inhibitor compete for access to the enzyme active site. This type of inhibition can be overcome by sufficiently high concentrations of substrate, that is, by outcompeting the inhibitor. However, the apparent Km will increase as it takes higher concentration of substrate to reach Km point or half of the Vmax. Competitive inhibitors are often similar in structure to the real substrate. The next is uncompetitive inhibition. The inhibitor binds only to enzyme substrate complex. This type of inhibition causes Vmax to decrease and Km to decrease. The next one is non-competitive inhibition. The binding of the inhibitor to the enzyme reduces its activity but does not affect the binding of substrate. As a result, the extension of inhibition depends only on the concentration of the inhibitor. Vmax will decrease due to the inability for the reaction to proceed as efficiently, but Km will remain the same as the actual binding of the substrate. The next is the mixed inhibition. The inhibitor can bind to the enzyme at the same time as the enzyme substrate. However, the binding of the inhibitor affects the binding of the substrate and vice versa. This type of inhibition can be reduced but not overcome by increasing concentration of substrate. Although it is possible for mixed type of inhibitors to bind in the active site, this type of inhibition generally results from an allosteric effect where the inhibitor binds to a different site on an enzyme. Inhibitor binding to this allosteric site changes the conformation of the enzyme so that the affinity of the substrate for the active site is reduced. And these are the examples of enzyme inhibitors which are produced from the microorganisms. Here the class of drugs is phytostigmine, rivastigmine, tacrin, diphlos, malathion, carbal, propax. These all are the acetylcholinesterase inhibitors, and the aspirin, ibuprofen, nimesulide, paracetamol. These all are COX inhibitors. These are used as NSAIDs, and anti-gout drugs are allopurinol, febuxostat. These are xanthine oxide inhibitors, and That the diuretics class of drugs are acetazolamide, dorzolamide, methazolamide. These are carbonic anhydrase inhibitors. Mirlonone and celestrozole are phosphodiesterase 3 inhibitors. Atorvastatin, simvastatin, lovastatin are B-hydroxymethyl coenzyme reductase inhibitors. Letrozole and anastrozole are aromatase inhibitors. Insulin is tyrosine kinase activator. Acrobose, miglitol, voglibose are alpha glucosidase inhibitors. And these are the some of the inhibitors that are produced from the microorganisms. Any doubts?
Naveen sir,
Madam?
it's completed, no? Shall I request for queries?
Yeah, if necessary, there are some more slides, but it's more than 1 hour now. Even I want to close.
Dear participants, if you have any queries, you can post it in the chat box, or you can directly interact. We'll allow to interact with sir. First of all, I want to thank Dr. K. Naveen Babu sir, understanding the emergency situation the other side, readily accepting within no time for readily accepting the request to carry on the session for today. Thank you so much, sir.
Okay, thank you, madam.
I'm not ready, sir. I request Dr. Varshinkar sir to give the concluding remarks of this session.
But I would like to thank Naveen sir for spending his valuable time for enlightening us with the topic on bioactive molecules from actinomas stress. Sir has given a clear-cut information and detailed information on both the source regarding the techniques for isolation, along with their advantages and disadvantages of each technique. What are the different media that are required for the screening, about the incubation, about the colonies, and also the subculture, and also the minimum requirements, and also the minimum what are the minimum precautions we have to take regarding the subculture that Sir has enlightened us, and Sir has given a detailed description. Sir has discussed in detail about the fermentation of the cultures, extraction of active metabolites, followed by the screening, and next, and with the determination of activity of isolated compounds, where the sir has discussed regarding the two topics like antimicrobial activity and enzyme inhibition. So thank you, sir. Thank you very much for your complete and detailed description of all the various methods. So how the product has to be extracted from a microorganism. Thank you, sir. Thank you for spending your valuable time.
Thank you.
I request the participants, if you have any doubt, you can ask. Sir.
Dear participants, I have shared the today's attendance form. Even in Telegram group also will be sharing. So there's small announcement for tomorrow's session. Tomorrow morning session is to be taken up by Dr. Falguni Patisar from IIT Hyderabad. Sir is busy in the morning schedule, so he has postponed the session to the afternoon one, like today. Sir will be taking the session from 2:30 onwards to till 4 o'clock. The total 1.5 hour session will be conducted for tomorrow. As it is indicated, 1.5 hour session. Instead of morning, we are going to conduct it from 2:30 to 4 o'clock. And this the same will be intimated in Telegram group also. So if you have anyone missed, through mails also it will be intimated. Okay. This is only the small change for only for tomorrow's session. Instead of morning, it will be taken up in the afternoon session. That's it. Thank you so much, all of you. We are concluding the session and we'll meet tomorrow in the afternoon session at 2:30. Okay, sir. Sir, I'm concluding the session, sir.
Yeah, okay, Rifa. Thank you.
Thank you, sir. Thank you, all of you.
Shall I end the meeting?